Dear students, today we are going to study lysosome, which is another membrane-bound organelle. It was discovered by Didier. It takes gomeri-stain for acid phosphatases, and the etymology of word lysosome is that in Greek, lysis is dissolution and soma means body. The lysosome is a membrane-bound structure for intracellular and extracellular digestion because it contains a battery of enzymes which is collectively called as acid hydrolases, and they digest most of the compounds which exist in the cell system or the living system. They are found abundantly in macrophages and the cells which are indulged in enzymatic actions, for instance liver cells, pancreas, thyroid, spleen, intestine and kidney cells. As far as the structure of lysosomes is concerned, they are spherical in shape and are single unit membrane bound structures having lipoprotein composition and dimension of around 0.4 to 0.8 microns and contains hydrolytic enzymes which are called as acid hydrolases. I had mentioned in another lecture about the labilizers and stabilizers. The labilizers are those compounds which tend to destabilize the membrane of the lysosomes, thereby facilitating the release of enzymes from the lysosomes. Such compounds are called as labilizers, and the examples are vitamins A, B, E, K, progesterone, testosterone and beta estradiol. But there are certain compounds which have stabilizing effect on the membrane of the lysosomes. For instance, cholesterol, cortisol, vitamin E, chloroquine, antihistamines and heparins. They have stabilizing effects, thereby they prevent the passage of the acid hydrolases out from the membrane of lysosome. I also mentioned that they exist in multiple forms, and the primary lysosome or the nascent kind of lysosome, when it is newly formed, it is called as primary lysosome. Whereas when it gets associated with phagosome or food vacuole, it is termed as phagolysosome or heterolysosome. And when it engulfs its own organelle like mitochondria, rough endoplasmic reticulum or smooth endoplasmic reticulum, Golgi body, for the purpose of growth and repair, then it is termed as autolysosome. After when the lysosome or primary lysosome digests the material which it has taken inside, the leftover body is termed as stylolysosome or residual body. The residual body contains undigested material. Then come to the chemical nature of the lysosomes. As I have mentioned that the lysosome, they contain many enzymes and they act upon certain substrates. So I am going to mention here the enzymes and the enzymes acting on certain substrates and their byproducts as well. First of all, the lysosomes have nucleases which act upon polynucleotides and converts them into nucleotides, sugar and nitrogen bases, phosphates and pentose sugars. In the nucleases we have acid ribonuclease whose substrate is RNA, and acid deoxyribonuclease acting upon DNA. The first one converts RNA into bases, phosphates and ribose sugar. Then The second one, which is acid deoxyribonuclease, acts on DNA, converts it into bases, phosphate and deoxyribose sugar. Now we have another class of enzyme which are called as phosphatases. These act on phosphate esters and converts them into monophosphates. These phosphatases, we have two kinds of phosphatases, acid phosphatases and acid phosphodiesterases. Acid phosphatases acts upon phosphomonoesters, whereas acid diphosphoesters acts upon oligonucleotides and phosphodiesters, converting them into monophosphates. The acid phosphatase also converts the phosphomonoesters into monophosphates. Then we have ligases. Then we have proteases. Then we have lipases, which acts on lipids and fragments them into smaller ones. Esterases acts on fatty acid esters, whereas phospholipases acts on phospholipids, converting them into smaller fragments. Then we have proteases, acts on proteins and converts them into amino acids. The examples are cathepsins, which acts on proteins and converts them into amino acid again. Collagenase acts on collagen, converts them into amino acids. Then we have peptidases, acts on peptides, converts them into amino acids. Then we have another class of enzyme called as glycosidases. The examples are beta galactosidases, beta glucuronidase, alpha mannose, and alpha glucosidase. These acts on polysaccharides and converts them into monosaccharides. Then we have sulfatases, which acts on sulfate esters and fragments them. So this is the chemical nature of lysosome, and they are actually the bodies which digest the certain biological compounds. Now come to the functions of lysosomes. First of all, it is extracellular digestion. They are involved in bone erosion. It is caused by osteoclasts. Dissolution of cartilage is affected by cathepsin D. Thus, they help in reabsorption and deposition of bone substances. Lysosomes are further involved in autophagy also and heterophagy. Digestion of endogenous material, for example, mitochondria, endoplasmic reticulum, or Golgi's, is affected by lysosome, and it is a very normal function, has important role in rejuvenation and renovation of the cells. And in adverse condition, when we get starved, the lysosome, they come into action and they start autophagy, deriving energy from the cell when food is absent. Then heterophagy, intake of exogenous material into the cell, and subsequently it is broken down by the enzymes which are present in lysosome, is termed as heterophagy. And pinosome and phagosome, they fuses with the lysosome. Pinosome, when it contains liquid material, the food vacuole is called as pinosome, and when the material is solid, it is called as phagosome. They gets associated with primary lysosome, and this is called as heterophagy. That means the material is from material has a exogenous origin, whereas in autophagy the material was endogenously, the digested material originated endogenously. The lysosomes are also involved in hormone secretion. For example, thyroid gland, it contains high molecular weight protein called as thyroglobulin. It is stored in a colloidal form. Thyroid hormone, thyroxine and triiodine are linked with this protein, and this protein has to be cleaved in order to make these thyroid hormone functional. The thyroid globulin enters the epithelial cell by pinocytosis and it fuses with the primary lysosome. The hormone split from thyroglobulin and released into the bloodstream. There, in this manner, it becomes functional. Then lysosomes, they play a very important role in fertilization. The acrosome contains high hyaluronidase, also found in lysosome. In the development procedures such as metamorphosis in insects, amphibians, the involution of uterus, mammary gland immediately after the birth, and the endometrial breakdown during menstruation, is all these phenomena, they are facilitated and effected by the lysosomes. Now lysosomes are also associated with certain disease. When there is increased autophagy, the disease is called as autolysosomal diseases. Intracellular release of hydrolysis due to change in the permeability is the cause of autophagy. It contains DNAs which breaks down the strands of the DNA, causes chromosomal breaks. The example is myeloid leukemia, which is blood cancer. It is caused by partial deletion of chromosome number 21, and this is brought about by lysosomal enzymes, DNAs. Now, the extracellular release of hydrolysis. What are the effects of releasing? If the hydrolysis, they are released outside the cell, then it causes the erosion of synovial cartilage and synovial fluid. By the cathepsin D, it breaks down cartilage, causes arthritis. Then lastly, we have accumulation of indigestible material within the lysosomes. It causes silicosis and asbestosis. Silica causes fibrosis of lungs, and gout is caused by accumulation of sodium urate crystals. So students, this is all in this lecture. So in the next lecture we'll be studying centrioles. These are actually the cellular organelles of microtubular organization and found only in animal cell, and during cell division they migrates to opposite poles and thereby ensures the separation of the replicated chromosomes. We'll begin it in the next lecture. Until then, thank you so much.